Hello. I want to stick my head in here and just introduce the topic for this lecture. We're having a general discussion about conservation, but I think it's really important to remember what is being conserved when we're talking about this. Biodiversity is a central concept to conservation, particularly in the tropics. So what do we mean by biodiversity? What are we measuring? What are we quantifying? How do we analyze it? How do we recognize it? So that's what I would like to discuss. And we're going to discuss first the morphospecies type, the phenotypic side of it.
One fundamental question we need to be thinking about is how do we measure biodiversity? Well, we do that by counting species, right? But then the question is, how do we then identify species? Species typically have unique morphological features that we recognize by looking at various populations of samples and specimens. And much of our basic understanding of tropical forest is based upon this very simple process of looking at specimens and understanding the variation in them. So what I want to focus on this lecture is the basic concept of morphospecies and how we morphotype things and measure biodiversity using that concept. Oftentimes we do this by looking at traits that are on the organism all of the time, like leaves or when you're thinking about plants. But one problem is that these morphospecies are typically based upon the personal opinion of just one or just a few experts. This can cause some problems and introduce bias. And oftentimes the morphotyping is never replicated. We don't do it twice, and we don't have a number of individuals do it separately. One real challenge with morphospecies is that morphology is often very messy, particularly when you're trying to capture standardized images for analysis. This can be very difficult to automate. There have been many attempts out there, like LeafSnap here. It's an app that you can download, but it's very localized in its geographic distribution, and still the photos have to be very taken in a very particular manner. Also, reference collections for identifying morphospecies are difficult to share, and they can oftentimes have very high overhead costs. Of course, there are other ways now to identify species like the barcode of life, but I'm not going to talk about that right now. So I want to talk about right now is how can we use technology to exploit the powers of the human eye in digital photography? So I'm sure you're all aware of these citizen science approaches like Zooniverse, which is a website where people can go and help identify galaxies and identify star formations and so forth, so they can really contribute to science. So I thought of this project last year at TiTAF, just taking fallen leaf collections and presenting a citizen science with a choice. Show them two images of leaves and simply say, are these two specimens the same species or not? Yes or no? A very simple question. And so that the citizen science could look at these and make a direct comparison of them and decide whether or not they're the same. So as I've mentioned, I want to revisit a study that I did in selectively logged forest in West Kalimantan, Indonesia, where we looked at the tree species diversity in these different types of forests after logging. And this is all based on morphospecies concepts and typing fallen leaf specimens. So what I want to do is open this up to citizen science approach, as I've shown in the TiTAF example, and just see if people can get the same results that I do, that the logged forest was not that less diverse than unlogged forest after just 8 years. So I've given you the same assignment to do this leaf matching. As you've seen in the folders, you have pairs of leaves compared together, and I just want a simple yes or no answer whether or not you think these two things are the same species. As I suggested, you should probably go through the folder and get an idea of the variation. But this will double my sample size, and we can really think about how to analyze this data and whether it's going to be accurate or not. And then I want to ask you several questions. Do you use this morphospecies concept in your own work? If so, how frequently? Does it play a critical role in the analysis? And then how might it affect your interpretation of the results if someone else did the morphotyping? Another question is the morphospecies valid, considering now that we now have powerful DNA sequence-based technologies that can do things very quickly and very efficiently and get very large numbers and really quantify things? A third question would be, given your organism of interest, do you think this type of citizen science approach could work for you? Could it provide a more robust and rigorous result than simply doing it by yourself or relying on experts? And question 4, how would you analyze this type of data? What kind of results would you hope to get from it? And would it really provide a better way of determining morphospecies than simply doing it yourself or relying upon experts? So that's all for now. I think there's a lot there to think about and discuss. It's very basic, but it's very central to our concept of biodiversity. And I'll be making a more detailed presentation about the selective logging study that we can then move forward with those samples. Thanks a lot.